Today we'll be presenting webinar 5, the API and the R client. So we've had 4 webinars so far every Thursday. The previous 4 were more focused on using the website, and this is really more for kind of advanced users that would like to programmatically access the data in the website. And this is the last webinar for now. So thanks so much for joining. It's been really cool to organize this. And if you have any feedback, please fill in the feedback emails that we'll send. And if you have any ideas for future webinars, let us know. So first, I want to a shout out to everyone who worked on cBioPortal. It's like a lot of institutions, the funders. For this webinar specifically, also want to thank Marcel and Levi that worked on the cBioPortalData R client. So and Christoph in our lab that also helped develop the workshop. So you can ask questions using the Q&A feature. So they will be answered, they might be answered live, some will be answered directly. There's a whole group of panelists that will try to answer them. The recordings will all be posted online at cBioPortal.org/tutorials. And yeah, if you still have questions after the webinar, you can ask them in our Google group. So you can email cBioPortal@googlegroups.com. Now, I'm using a big screen here, so if you can't see the full screen, I recommend that you click at the top at View Options and then click on Fit to Window so that it will scale to your display size. So, well, as you know, this is the 5th webinar, so cBioPortal, is this platform for interactive visualization and analysis of cancer genomics data. Everything is open source. You just saw all the different institutions that are involved with developing it. There's a public website, cBioPortal.org, with public data, which is what we'll be using today. And there are also many private instances installed all over the world, like at academic and commercial institutions, and they have their own data loaded into their version of cBioPortal. So another thing that you can do on the website is you can make oncoprints and lollipop plots by submitting your data. And today we're also going to add that and explain how to actually make your own visualizations using public data. So what you will learn today, I hope at least, is what a REST API is and why you would want to use one. The second thing is actually how to use the R package, cBioPortalData, to pull data from the API. And there's a lot of stuff that we actually will not cover because obviously we only have about 45 minutes, so there's only a limited number of things that we can talk about. So things that we will not cover are the basics of R. So I'm going to assume you know a little bit of R, and ideally you would know something about the tidyverse as well. This is like a set of packages that are related to data transformation and plotting. We also will not really be talking about the introduction to Bioconductor. So cBioPortalData is an R package part of the Bioconductor universe, and these are kind of like a set of packages related to biological data analysis. So all these different packages, they have great resources that you can find here, and you can read up more about it. So I recommend you to do so if you're not familiar with that yet. That being said, we will continue. So if you're still here and still interested, so then I'll just briefly explain what a REST API is. So we'll break it down. So the first thing is we're talking about an API. So that's an application programming interface, just sort of like a set of rules by which one application can interact with another. And that's maybe kind of a vague description because it is in general spans a lot of different things. You can imagine if you write a function in R and some other program calls that function, you sort of have an API. This function is sort of an API. You could see it that way. So what is the REST part of REST API? So REST stands for Representational State Transfer. That's an architectural style and approach to communications, which is often used in web services. So that's a mouthful, but in short, it's really just a client and server that are communicating with HTTP requests, and they send data in JSON format if they send data. So here's an example. We have a post request from a client. They send over this structure. So this is a JSON format. So it's not too complicated. It's like, you see some sort of array here and then a map of key to values. And that goes to a server. So it goes to a particular address, a particular URL here. So /server/weather. And that server then responds and it sends back another JSON object. So low 16s and high 23. So this is like how a large part of the internet communicates, how a large part of different servers communicate with each other. So they're really used everywhere. So you can go to, for instance, google.com, and if you open that in your browser, if you use Chrome, you can go to click on View, click on Developer, click on Developer Tools, and then they will open something like this. And you can actually see, if you go to the Network tab, you can see the particular HTTP requests that are being sent. And so one of them here is being sent to some endpoint, some API, and it returns a JSON response. And in this case, it's kind of showing all the different things that I searched for recently. So that's just an example. So you can see kind of how ubiquitous these REST APIs are, that it's really all over the place. So now an introduction to cBioPortal's REST API. So you can find that here, cBioPortal.org/API. So the first thing you'll notice here is that it's in beta. So we're very close to officially releasing version 1. It's pretty stable already, but just keep that in mind that some things might change. So when I click on this, I get here. So I'm just gonna blow up my the font size a bit so you can see it a little better. And when you go to this page, you can really see all the different kind of objects that we have here. So you see cancer types, studies, patients, samples, a whole bunch of different kind of models or objects that we have in the cBioPortal world. So there's, for instance, a group of cancer types, and then you see there's what is called a GET request. So this gets all cancer types. So you can see there's a bunch of different parameters that you can pass. So I really just clicked on this. You can, for instance, click on try it out. And well, we just use the example, for instance. So you can see for curl, this is a command line tool. So you could, for instance, copy and paste this on the command line and you will get a response. You can also just open this with your browser, since what your browser does, it just sends an HTTP GET request. And so you can see here what the response is. So you get like an array, and each object is a cancer type. So that's pretty obvious now, I think. So then another example is, for instance, studies. So we have a lot of different studies. When you go to the homepage, you see a listing of all the studies. So here we can, for instance, there's an endpoint to get all studies. There's an endpoint to get a single study. So you can, for instance, try, all right, let's try to get this ACC TCGA study. So you'll get a response like this. And again, you can also go with your browser there and see something like this. So I have installed some plugin in my browser that nicely formats JSON. Like if you don't have that installed, it will just kind of like show it all after each other. But you might want to install something like that. So it's called JSON formatter. So that's just kind of giving an overview of the API. And then, so you saw these GET requests. Now you also have the POST requests. So with the GET request, you see we're not really sending JSON data. We're only really going directly to this URL. With POST, you can fetch some by one or more IDs. So you can kind of see here, you can only get one study. But if you want to send a single request to retrieve, for instance, 2 or more studies, you can use this. So say we use this study again, ACC TCGA. You can also send in 1, so we'll just do 1 for now. And you'll see that the request that you get does something like this, and it adds this little JSON part to it. So I hope that's visible. So yeah, that's that structure, and it does some escaping of the quotes here. That's just because if you copy paste it into your shell, you need those quotes to be escaped, and you'll see you get a similar response. So that's really the core mechanism here. Like GET is usually for like one object, and then for POST you can retrieve multiple objects in a single request. And that's not the case with all APIs, but that's just how we designed ours. All right. So now the question is, why use a REST API, right? So now I kind of showed you what it looks like, but why? Why would you actually use one? So there are a lot of different reasons. So one, it's flexible. So the fetch data that you get, you can analyze in any way. So on our website, you might be limited by only, the features that we have. Although we have many, we don't have all the different use cases. So this way you can kind of further analyze it. And then there's also, it's structured data. So as you saw, it's like, objects that are in arrays, so it takes little work to transform them. Whereas you can imagine if it's like more some garbled text file, it's a little harder. You can slice data, so you can only get what you need. You don't have to download all the data just to, look at a few genes or a few samples of your interest. So you can really request just those that you're interested in. cBioPortal has a lot of curated data, so we have a whole team of curators that that looks at different studies and imports their data. So that's another reason why you would want to use the REST API, because you get access to all that data. If you develop some sort of automated process, this allows you to you don't have to do any manual steps to download the data. Then the other thing is the API changes a little over time. So we version the API, and this, will make it very clear when there is some model that has changed or something else, and we'll, have backwards compatible support for older versions. So the other thing is it's reproducible. So if you make some analysis, you can easily rerun it against updated data, or maybe you want to run it on your private data. So maybe you have a cBioPortal instance locally at your institution, and you want to run the same script, or you maybe want to combine data from your private instance with the public instance, so that you can all do. The other thing that's neat is that you can reproduce the exact data pooling that happens when you visit the website. So what I mean by that is, like, you can actually go to cBioPortal.org and find some, maybe you look at the OncoPrint or one of the other visualizations. You can then click on the network tab and figure out which API endpoints it's calling. And we will show that when we get to the coding part of this webinar. So some more example cases. So reproducible analysis for a paper. Maybe you want to build kind of a custom dashboard. You want to do a custom kind of visualization or analysis that's not yet in cBioPortal. And the other nice thing is you can run performance heavy analyses. So you can imagine for our website, we try to keep it as stable as possible. But so some type of analyses are, they're very performance heavy, and we can't really support them for all users. But by you downloading the data and running the analysis locally, you can actually do those types of things. Now, there's several ways to download data from cBioPortal.org. So the first way is manually. So you have these plots on cBioPortal. They often have like a download option that you can click on, and they allow you to download the TXT or a TSV. So this is good for kind of a one-time quick thing. You maybe want to adjust a small thing. So that works pretty well for that. But, although we try to keep, the data formats the same, it's not, versions like the API is. The other option is you can download the data from DataHub. So here we keep all the curated files that we load into cBioPortal. So these are like TXTs and TSVs. And sometimes this might be useful if you just want to download all the data, right? Like you want to pretty much just have everything. The downside of using that data is that transforming the data is a bit more work. So although it's TSV and TXTs, you don't really get, the nice objects that you have when you use the REST API. And as a side note, I should say that the cBioPortalData R package does provide some interface to download this data directly, if you prefer to do that. And then lastly, what we'll be working with today is the REST API, and this is really for all other cases. So this is really the recommended approach if you want to, do anything else than what I just mentioned. And I should note here that it's also fine to use for really large data sets, because it does local caching. So it basically, when you pull the data, it will store it locally on your machine and won't repool it again. So that's a really nice feature, because you can, that way, just kind of, you don't have to worry about storing the data in some way. If you're using, another client that's not R, then maybe you first want to have some script that downloads all the data to your local machine, and then you have another script that actually does the visualization. With the cBioPortalData package, you don't really have to worry about that part. So now, how do you actually use it? So the first thing is the documentation. So we have two links here at the top, Web API and R MATLAB. So they're all pointing now to docs.cbioportal.org. So that you can see here. So there's explanation on the API and API clients. And so we have some explanation on the R client. There is the Python client. And there are links to the workshop that we're doing after this, like the coding exercise. And for Python, we also have some workshop that you can find. But really, all kinds of languages are supported. Like you can do JavaScript or TypeScript, whichever you're interested in. So you can use this tool, the Swagger CodeGen, to generate a client in kind of any language of your choosing. And so you're not really limited to the ones that I just mentioned. So now let's talk about R specifically. How do you use the REST API in R? So there are two R clients. There's the CGDS R. So this we've had for a long time. This will unfortunately be deprecated because it uses an older version of our web API. It's on /webservice.do. So although it still works and we'll keep it running for a while, you really want to start migrating to the new client, the cBioPortalData client, to access kind of all API endpoints from the new website. So the one that I just showed, the cBioPortal.org/API. So that's one of the things that this client offers. So on top of that, this client offers a lot more. So there is the Multiassay Experiment interface. So it provides the multi-assay experiment interface. So this is an interface that's kind of part of the bioconductor universe, and it's a very popular data structure to kind of handle multiple assays, like mutations, expression, methylation, and you can kind of easily subset and reshape data using this data structure. And like I said, it also has this kind of local caching. So when you use the REST API, it will actually save the files locally, so it won't repool them every time. And yeah, so this is very convenient. So I won't really talk too much into depth about how to use multi-assay experiment because it's a little too much for this webinar, but I encourage you to kind of read up on how to use it further. So now we can go to the coding bit. So this is a link to a GitHub repo. So yeah, you can either just you know, look what I'm doing, or you can go here yourself and check it out. So this directly takes me to the GitHub repo, and you will notice, so there's a workshop here in HTML form. So you can kind of read it like this. All right, you can just kind of scroll through, but also, so all the code for the workshop is in here. So you can also make changes and, you know, play around with it. But also I want to add, so you can actually run this directly on mybinder.org. So when you click on this button here, it actually starts a machine in Google Cloud for you that you can control through your browser, and it will boot RStudio. So this is kind of one of the most common development environments for R. So yeah, you can look at that one. I already installed RStudio locally, so I'm just going to be walking through here. So kind of how this works. So here on the right, you see the document that I was just showing. So that's basically the web page that it generates. And here on the left is kind of the raw code. So this is called R Markdown. So you can combine kind of text things. Like you write a header like this. For instance, we have introduction there. And you can combine it with code blocks of R. So you can, for instance, run this by pressing on the play button. And that will actually run it. And so, yeah, I have a section here on installation. So like I said, the easiest way is to really use mybinder.org and click on that link. So once you've done that, let me see if this already started. This is still running. So it takes a while before it starts. But once it starts, it works pretty well. So this is what you maybe want to do if you want to look at it now. I also recommend after the workshop, you know, you can kind of, you can install it locally. So if you're familiar with R and RStudio, you can easily install cBioPortalData like this. If you don't have R installed yet, and maybe not RStudio even, you could also use Bioconda. So this is another way to install the software. So there's some explanation there on how to do that. So this is all, you know, afterwards, I kind of recommend that you look into this and figure out what the best way is to install. There are also Docker images and Amazon machine images, if you know, if that's what you're familiar with. I would, from these ones, just pick whatever you're familiar with and install it that way. So now that the tedious part is over of the installing, so as you can see, hopefully it wasn't that tedious because you can click on the link. So this is in my browser now, right? This is not my local machine, this is in the browser. So I can actually click on this workshop file, and then you can open that here. And now you can start running these commands. So you see when you press play here on one of these code blocks, it will actually run it in the console. And same here. So this will take a while the first time that you load it. So this is really saying like load these libraries. But after a while you will see that it will have loaded those. So now we can start really with how to initialize the API client. So let's get to this section. So in the Initialize API Client, I'm gonna go and look at the code here. So that's this part. So I can run this, I press play. And then, so as you can see here, it generates what it looks like if you print that object, the cBio object. And what you can see, you see all these different groupings that we saw before, the studies, the cancer types, and then the operation and some explanation of what it is. In RStudio, you will also see this table. So you can actually navigate through it like that. And right now my screen is like divided in half, so I only see one column. But if you use it full screen, you'll see a few more. So this is kind of one way you can also look at the API. So one of the things that you can do is that it has command completion. So it also says here, use cBioPortal $tab completion. So you can, for instance, type studies here and you will get all the studies related endpoints. So that's kind of an easy way. So I just press the $ and then I type studies. And then you get this whole list and you can, select any of those. So that's one way you can kind of navigate the API. Now, to kind of use it, figured we compute some simple stats. So here are some. So maybe the first one, how many studies are there in cBioPortal? How many cancer types do they span? How many samples are there in total? Which study has the largest number of samples? So some questions like that. So the first thing is always you want to find the relevant endpoint. So which endpoint should you actually be using to get this information? So for the first question, how many studies are there? There are a variety of ways you can accomplish finding that. So we already saw, I typed in the $ and then did studies. That's one way. So I can just try these. That would be one way to do it. The other way is you can use the API documentation. So I briefly showed that before. You can go to the website and kind of navigate and maybe click on some, like you try out, you press the execute button and you look at the response. So that's another way to do it. And then the third and last approach is using the browser development tools. So that was kind of the same as what I was showing with google.com. You can do the same thing on cBioPortal.org. So I just want to show that once because I think this is kind of a really kind of neat way to figure out how some of these visualizations are done if you like just have no clue where to start, right? So you, for instance, see this section cancer studies, right? So I can click inspect or here view developer tools. And I go to the network tab here. I see nothing now because the requests have already been fired. So I just have to refresh the page and make sure you click on this one and network, and then you can filter for API because I know everything is going to be cBioPortal.org/API and then something. So I already see here there's 3 endpoints that it's using. So I can, for instance, look at this. Let me just blow this up a little bit. So maybe it's too much. So you can, for instance, click on this one, and what you'll see, you'll get this response, right? So all the studies are here. So this makes sort of sense. The name of the study, the cancer type ID, and we see that there are. Let's see, can we go back? Yeah, so you can see there are 285 elements. So it says 284, but it starts with 0. So there are 285 elements in total in this object. And we can see here that the portal contains 285 cancer studies. So that indicates that that's probably the right endpoint to use. So once you know that, so I'm just gonna go back to my local machine. So once you know that, now you can start pulling data from the endpoint. So this is always kind of the first step to actually figure out what endpoint to use. Then, so you got 3 approaches to endpoint searching. So now we can actually get the data. So if we use this, we can run this again. So you get a response object. So this is an HTTP response object, and you'll see there's a status of 200. That means it's okay. There are a bunch of other statuses related to HTTP. 200 means success. So in this way, we still can't really check how many elements are in there. So there's like a package HTTR that you can help to parse this response. So then you get it into an object that you can actually look at more easily. And then you'll see the number of elements in the response are 285. So like I said, we know that each element represents a study. So we can now answer that first question, which was how many studies are there? And so we see there are 285 studies in cBioPortal. And we already noticed here, for instance, I took a screenshot a few days ago that was from 284. So you can already see that there's a nice advantage to using the API because you actually get the live information instead of static, like with my image here. So now, as you see, there were quite a lot of steps involved, and we had to manually parse the response, and that was a little tedious. So cBioPortalData actually has all kinds of convenience functions that do this for you and that make it a lot easier. So instead of having to figure out the endpoint for studies, you can actually simply use the function getStudies. So there's a getStudies in cBioPortalData. You can run that on the client. So you pass the client to this function. And then you get a nice table back here that has all the information that we need in a table structure. So you see there's 13 columns. You have the name, description. So this is much easier to handle in R. And this kind of object is actually called a tibble. And that's part of the tidyverse. Yeah, it's like a more refined version of the standard table object in R. So now you can easily get the dimensions of the table with the dim function, the dimension function. So you can press play. You can also do this faster by pressing, if you're on Mac, Command-Shift-Enter or Control-Shift-Enter if you're on Linux or Windows, I believe. So then you see there are 285 rows. So that corresponds to the number of studies in cBioPortal. So you can again do, there are 285 studies in cBioPortal using this studies object. So this was much easier to use than actually directly calling the API and parsing it ourselves. We have this nice convenience function to do this for us. Now we can also take a look at what columns there are in this table. So you can, for instance, do call name studies, or you can browse it through this thing, whichever you prefer. So for the second questions, we wanted to figure out what cancer types they span, right? I believe that was it. Yeah, how many cancer types do they span? So there is a column here. Let's see, it's related. It's called cancer type ID. So you can actually, run this, and you will see, you can basically do a unique on the number of studies on the cancer type ID column, and then get the length of it. And then, you'll see there are 88 cancer types. Now, there's also a column called all sample count. So that has the number of samples for each studies. So that's here, and that's the third question, how many samples in total? So you can run this, and you can see there are 86,830 samples in cBioPortal. And then for question 4, the studies with the most samples. So then we just do a max on that particular all sample count column, and we find a study with the most samples is the MSK Impact Clinical Sequencing cohort from 2017. So how could you have known actually that in the beginning that there was this function getStudies, and you wouldn't have to go to all the trouble of parsing the API responses? So there's a function R to list all functions. So you can run this, and this lists all functions in cBioPortalData, and you will see there's getStudies. And Additionally, you can also do something like this. So you can, for instance, check, you can use tab completion. So this kind of, do this. And now, you can search for studies and you will see that there's an object actually already. You don't even have to call get studies. So that's another way to do it. You just use tab completion to kind of get an idea of the different functions and things that are there. So there's also a more user-friendly page that has all these functions. Let me just scroll down here a little bit. So we have to get studies. So there's a website here that kind of explains cBioPortalData. And another option, this is very common in R, all packages kind of include this vignette. So you can, this is like a manual to your package. So you can, for instance, do browse vignette, and you can run that. And then you can find some of, like, for instance, the quick start guide for cBioPortalData, and it has all the information. And usually there's also, you usually can also find this on the website, but if you're already in R, then maybe you want to use this command instead, if you find that easier. It's kind of a convenient interface because if you know the package name, you can easily find the documentation. You don't have to like Google and stuff. And I have to say that this doesn't work on mybinder.org. This only works locally. So yeah, in general, it's like good practice. You first want to check, is there some function or some object in cBioPortalData that pulls the data that you need? And if there's not, then you can actually go back and resort to parsing the API responses directly, as I showed. So just a quick, visualization. So say we want to recreate this cancer studies plot. Well, the first thing you usually have to do is transform the data. So this is kind of maybe a complex statement, but it's like this is using the tidy universe to kind of transform it. And once you get familiar with it, it's actually quite convenient. So this is the studies object that we had before. We group by the column cancer type ID. We get the sum of all the samples, and then we're gonna store that as total samples, and then we're gonna sort them by the total samples, and we get the top 20. So we run it like this, and that's being assigned to cancer type count. So you see this kind of weird character. So this is like a pipe character. It sort of just means send the result from this statement to the next function. And now we can also see what this object looks like. So you see the cancer type ID and the total number of samples, and it's sorted. One thing you'll notice, it says mixed here. We don't really see that in this plot. We don't have mixed cancer types. So these are studies that have, a mix of different cancer types. And so we can easily filter that one out. So we add some statement, filter everything that is mixed. So let's do that again. So now we don't see mixed anymore. And now we try to plot it. So this is using kind of bar plot and a few other tools to plot this. So, I mean, I don't want to go into too much detail on how this works, but you can look this up later and play around with it. So this is a very bare bones version of what we saw here. And of course, you can actually try to, so we see here the cancer type IDs. You can actually use the cancer types endpoint to get, the full name of the cancer type. And yeah, and then you can find nice colors, etc. Yeah, another thing here is mentioned, you can use the detailed projections. So in our API, we use different projections. So you can do a summary projection of an endpoint or a detailed projection. And the detailed projection often includes other objects as well. So you don't have to do multiple calls. So instead of calling cancer types another time, you can actually include that by using a detailed projection. So then I just want to point you to some more examples that have been developed. So Christoph in our lab worked on this, and this really, kind of reproduce some of the charts that you're familiar with from a study view. So you can find that here. So it opens this, and we'll also convert this to R Markdown so you can easily use it. But it's just to give an example of how you can actually use the API and reproduce some of the study view figures. And yeah, there's a lot of other visualization resources. So, aside from the bar plot that I showed in the plot functions, there are many. So cBioPortalData is really more about getting the data and then the next step is the visualization. There are other packages to do that. And yeah, the tricky part, is usually kind of transforming the data to work for that particular tool of interest. But the nice thing is, like, once you've actually done it, you can easily reuse it every time. So that's kind of really where the power of using REST API comes into play. So really briefly, I'm going to add another, just, what is multi-assay experiment? There's a section here. You can go here to their website and read more about how the structure exactly works. But basically, what you'll find is, like, once you start pooling a lot of different data, different assays and clinical data, it kind of actually becomes complex to manage all that data locally. So you want some sort of nice data structure to easily subset into that data. So that's really where multi-assay experiments shines, and you can really easily combine multiple assays. So here you can see mutations, RPPA, methylation, and kind of have that all in one spot. And there's an example here that follows kind of some of the manual for multi-assay experiment and applies it to cBioPortalData to make some of these plots and survival charts, for instance. So yeah, then I would just say, for further reading, like I said, check out cBioPortalData website, the Multiassay Experiment, and yeah, more general, the R and Bioconductor, like there's a Bioconductor workshops, and there's the tidyverse as well. And I think I also just see that Levi and Marcel might give another more in-depth webinar on using multi-assay experiment. So yeah, the webinars are being made live. Very cool. Okay, yeah, I think we can go on to questions. We still have 15 minutes, so I think I can answer a few questions. Oh, wait, one more thing. I have a few more slides, actually. Let me just go over this. So I want to add, if you want to use cBioPortal for publication, use it. It's awesome if you use it. We're very happy if you use it. The studies that you generated the data for, please cite them as well. It's very important that the data gets generated. So of course we want to support that. The cBioPortalData manuscript, so the R package is still in review. But once it's out, please cite that as well if you're using that. So we can, continue development on this client, and also cite cBioPortal as indicated here. We would appreciate that a lot. And so, yeah, if there are further questions, so the recording of the webinars will be posted online, and yeah, if you have questions, you can email them, and you can use the Q&A. I will go over a few now. Thanks a lot for joining us.
All right, thanks, Eno. So looks like we have a few questions, and I think questions are still coming in. So if you have any more questions, please type in the QA session, and we will try to answer them either live here, or someone will type their answers. So let's see. And so it looks like people really like this webinar series, and a couple of attendees asked for future possible webinars. The first feedback is that potentially in the future we can divide webinars in levels, like beginner, intermediate, advanced, so that we can have more time to explain things deeper. That's a great suggestion, and we will consider that when planning future webinars. The second one is someone asked if we can have a cBioPortal local installation webinar. We're actually thinking of having a webinar on that, and we will announce it hopefully soon on our website, so stay tuned. The third one is someone asked if we can have a multi-assay analysis in our webinar. I know that Levi and Marcel, they're actually doing, previously they ran some meetup on that topic. So we will work with them and see if they do any more webinar, and we can announce that in our channels, such as our tweet or user groups. So there's another one actually just come in. Could there be a workshop for cBioPortal's top 5 use cases, which are complex? I think that's also a great idea. It's similar to the previous question about dividing webinars in levels. We will consider this in future webinars. And the second question we have is, where can we see the source code for what you've demoed in R today? And there are a couple of questions about like, how can I get links, etc. So we will share the link on the cBioPortal website, and we will also send the link together with the recording in a follow-up email after this webinar. The third question we have is, the API currently is in beta. What does that mean? And when will be the official API release? Can you say a few words about that?
Sure. So we're really just kind of finalizing and fine-tuning the last couple of things. So I think there will not be any major changes anymore. There's just a few things that we want to make sure that it's really proper and future-proof. So I think you can already start using it, and most endpoints will stay the same. Like it will be very minor change, and I doubt it will affect your analysis.
Another related question is, you mentioned that the API rarely has breaking change. Can you explain how the versioning scheme we use can help indicate the new version of API that has breaking changes?
Right. So this is actually one of the things that we're trying to finalize. So how to exactly version it, but we will probably follow semantic versioning. So that means you have 3 digits. So you have 1.0.0. The first is the major, the second number indicates minor, and the last indicates patch. So all the patch changes, so the last digit changing, that wouldn't really break anything. That's just bug fixes. And then the minor change is usually some backward compatibility change, some backwards compatible change, like a new feature or something like that. And then a major version, that's the first digit if that changes. But yeah, we will, once we release the first version, we'll keep supporting that API. Even, if we change major version, we will keep running an old version as well. So your analyses won't be affected.
Okay, so the next question is that the API work with our version below 4.0. I see that Levi and Marcel answered this question. We will not be able to support our version below 4.0 for the bioconductor package, especially if you want to implement multi-assay experiment. But I assume that for any R, they can connect to the API directly.
Yeah, that should be fine. So on the website, it also mentions the R API client. So there's another one. I can just briefly show that. So when you go here, so I mentioned in the slides there's 2, but there's kind of a third one that you could use. So there's this one, our API client. So this is what cBioPortalData uses under the hood as well. And that one, I believe, is compatible with older versions of R as well, as far as I know.
Okay, so another question is how to access through API in a private instance that requires authentication.
Nice. Yeah, that's a very good question. So we're actually working on adding stokin support, and that is almost done. And yeah, we'll follow up in the documentation on how to use that exactly with the cBioPortalData client, because obviously this is a very powerful feature. Yeah, it will be out soon.
Okay, so I think we have let me see, we have a couple more questions coming through. So cBioPortal have the same samples between multiple studies. Do those samples have the same name in the returned data to detect them easily?
Yeah, that's an excellent question, actually. And the problem is actually more complex than just having the same name, because sometimes there might be, studies, they take a sample and they name it differently. So overall, we do provide a listing on the website. So there's here a curated set of non-redundant studies. So there are no overlapping samples there. So you can try and use those studies if you don't want to have any overlap. Yeah, I'll add some documentation about how you could use this. But that's an excellent question. And so in short, the sample names might be the same. So for TCGA studies, they're probably the same, but there might be some studies where it's not the same. So we kind of manually figure out which ones are the same, and have a curated list of which ones are non-redundant.
There's a question about, let me see, it's not related to API, but are you planning integrating data from PDX data sets, such as the PDX Finder? Maybe I can take this. So we actually, we talked to PDX Finder a couple of years ago, and we're still considering, basically working with them and pulling some of the data from their resources. It's still in the to-do list, so potentially in the future, yes, we can actually hold some PDX data as well. It can be also a good future webinar series, so that other people and from the community, people can actually contribute data to our data hub that Eno demoed. Let me see, other questions. Could you please show more API query examples from a variety of other endpoints, please?
Yeah, so for that one, I would really kind of recommend to look at some of these examples here. So this one, for instance, there's a PDF here that really goes more into detail about what other endpoints to use. So I would advise to use that. And then there's another example with Multiassay Experiment. I mean, I can demo some more, but I think it's easiest if you kind of look over these, right? And you can also always use the website. So you can, for instance, if you go to, let's see, we can do some query. This one, for instance, so this is a little more complex. You can use this one and kind of see how this data gets constructed, right? So you do /API, see fetches studies. So you see there's actually quite a lot of different endpoints that are being pulled. So this is definitely a good example also to use the multi-assay experiment interface, because it will actually kind of combine all these different endpoints and you don't really have to think about it too much. Yeah, so that's basically, I can't really show that many, so I would say try to click on the links and dive more deeply into it.
Yeah, I just want to add that, if anything you see from the cBioPortal website, you should be able to get it from our web API. So it's a good idea to explore the website. And then, if you see any useful data you want to pull, you can try to get it through the API. Yeah. Another question is, are you going to actively incorporate future cancer data? I think I can answer that. So we are actively curating data sets from published literature and also from other resources. For example, we're currently enhancing our data set from the TCGA Pancake Atlas. We're also curating the data set from ICGC. and we are monitoring the literature, but if you see any data set that potentially we can add to the cBioPortal, please contact us through emails. You can also create issues directly on the GitHub, on our Data Hub page. Let me see. So there's another question. Is there a way to interface directly to other bioinformatics tools, such as CyberSort? You know, I don't know about CyberSort. Do you aware? Are you aware?
So I am not familiar with CyberSort, but it is a really good question. So interfacing with other tools. So you can pretty easily interface with other REST APIs, for instance. So we also have OncoKB, they also have a REST API, so you can connect with that one. You know, Ensemble has a REST API, so you could interface with that one too. So really the possibilities are quite endless.
Okay, so let's take one final question. How does cBioPortal scale with very large data sets, such as the AUC Project Genie?
That's a good performance question. So it works pretty well overall, I would say, but we are still trying to make more optimizations to kind of handle, you know, hundreds of thousands of samples. So as you could see in the public portal, we had like 80,000 or so, but in some of the other instances, we have even larger data sets. So we can handle it, but yeah, there is still some room for improvement.
Great, thanks, Eno. I think we can end here. Thanks everyone for attending this session, and please stay tuned for future webinars.
Yeah, thank you. Thanks for listening.